So this morning I want to try to touch on 4 points. Each of them will be quick since Martha's made sure I go quickly, and I only take my allotted 17 and a half minutes, right, Martha? That's okay. So I want to touch on the life cycle of an unbiased metabolomics study. Sometimes these studies, if those of you who do unbiased metabolomics, sometimes those of us in the lab have a different idea of when those studies might end than the investigator or the PI or our collaborator. And so I want to talk a little bit about the life cycle of a study. Then I'll show an example of an investigation of the activity of a novel fatty acid synthase inhibitor as an example of that. I'll touch very briefly on the importance of QC metrics for a biomarker study, and then give you an introduction to one thing Waters is introducing, which I think is a really novel and excellent potential for a DIA strategy, especially for lipidomics. So I think most of us in this room probably know this, which is that when we're doing an unbiased metabolomics study, we start with our study design and LC-MS acquisition that occurs in little green boxes as outside of Progenesis. And what I'm showing in blue are for the rest of this kind of workflow are things that are handled within the Progenesis software. We use that for. So we import, align, and quantify the data. We can do some principal components analysis, percent CV, outlier removal at the sample level and at the analyte level. Kind of quality control our data. And then really the goal is to do some statistical analysis, and that can either occur within Progenesis, or often when we have complicated experimental designs, we'll hand the data off to a statistician. So that's in the little orange box at the bottom. It can be using export tools, just basically export the data out. So at that point, really the idea is to try to understand what those compounds are, okay? And so we actually insert another step within this pipeline, and we like to validate the quantitative information that we receive in Skyline and visualize that data for a couple of reasons. So I know a number of you may use Skyline. You may not really understand where Progenesis QI and Skyline, where the handoff is, and so how we use that in our lab, I think these tools are completely complementary, right? So what we do is we need to know what we're going after in order to analyze the data in Skyline. So we'll use Progenesis to basically do all our peak detection, quantitation on everything in the sample, and then we can statistically select those molecules, then validate that quantitative information for peak shape and all of those things to make a publicly accessible file in Skyline. And then we can basically, if it passes that check, we go on to do our putative identities. So I got to slide my workflow over a little bit, so we're going to move it. The next step then so once let me just go back here. So once we're here, sometimes depending on how much we have in our database search, how well annotated our data, we might be done. How many people like to be done at this step, right? We do a putative ID, we hand that off to our collaborators, they can potentially do some biochemical interpretation, and then we're finished. So at some point though, we may actually need to purchase or synthesize standards in order to validate those identifications. Obviously that needs to happen outside the software. And once we've done that, we then like to go back into the pipeline and do accurate mass and MSMS alignment to confirm the identification to the database. Okay, so this is a handoff. And then after we're done at this step, we feel much more confident in our identifications. This is one of my pet peeves about metabolomics, because it really is the case, still the gold standard for confirming an identification is running an authentic standard of that type and confirming accurate mass, retention time, and MSMS fragmentation. Our final step often is that we would, if it's an important molecule, we'll build a targeted method, and we'd like to run it on triple quad across a much larger number of samples. Obviously, we'd like to export those molecules and build a targeted method again in Skyline. So this is our workflow for our targeted or our unbiased metabolomic work. Sometimes we like to finish here, but we realize there's a lot of work that can potentially be done downstream. Okay, so first quick anecdote. We have been working with Timothy Haysted's lab at Duke. They might synthesize small molecules and they're in pharmacology and cancer biology, and look at the function of those molecules. So they recently published a novel fatty acid synthase inhibitor. Basically, they discovered this molecule using flex screening approach. They identified 13 active candidates, which they narrowed down to one based on how they select molecules based on the expected interactions in the body and sort of other characteristics. Then basically they confirmed in vivo that it blocks acetate and glucose incorporation into lipids, and they confirmed that this compound inhibits the purified enzyme. And so at this point they came to us and said we want to go to try to publish this manuscript. We need to know a little bit more about mechanism, and we'd like to do a lipidomics study to understand what is happening to the lipidome of the cells as a function of this fatty acid synthase inhibitor. So our job at that point then is to take the cells that they generate, plus minus drug. We do the cell disruption, a Bradford assay, to normalize to the total biomass between samples. So this measures protein, of course, but it also we use as a surrogate for biomass. So we normalize prior to doing the analysis, the lipid extraction. Okay, and then we measure this with acuity, UPLC, and a SYNAPT. And so one thing I'll say here is that we often do use DDA for lipidomics. And the reason for that is pretty simple. It's not because we don't like HDMS/E. It's the fact that we've found that there's some advantages with respect to having accurately isolate that precursor to know the common fragment. In lipidomics, of course, there's a number of common fragment ions that are generated using different classes. And it's very useful to know that fragment, that characteristic class identifying fragment ion, came from your precursor of interest. So that's kind of why we often use DDA. So kind of keep that in mind when I go to the end of the slide, the talk. So this is kind of an interesting way to show the complexity of this data. So what I've just shown, and we'll come back to this in a moment, but this is looking at retention time and accurate mass, mass to charge. And a lot of people will plot the intensity as a color here, but what I'm actually plotting is the significance of the change between these conditions as the color. And the size of the dot is the number of identifications we were able to get for this compound, right? So one thing you'll notice is there's a lot of really big dots, meaning no change with identifications or putative IDs, but there's also a lot of very important red dots, right, with very few identifications and putative identifications. So we know we're not getting everything, but we're trying to use all the knowledge we have in the chemical information and libraries to move forward as quickly as we can. So a couple of screenshots about the things that Progenesis helps us to really do quickly and easily. So first thing is that with every study, we run a study pool QC. And so this is a biological pool of all of the samples. So we take the 10 samples in this case, we make a pool of them after extraction, then we run that sample to equilibrate the column, and especially we run it then beginning, middle, and end of the study, okay? We can see that those study pool QCs cluster tightly together in the middle. So this study pool QC allows us to measure the stability and the percent CV of the biological average of all of those samples. And it allows us a check for every molecule, so the stability of every molecule and the ability of that, how well those markers are sort of behaving in the mass spectrometer at that point. So we do this for every study we run, be it metabolomics, proteomics, targeted, unbiased. We make the study pool QC and analyze it. The second thing it does, which is really nice, is it checks for linearity or bias in the quantitation. So if this is actually the biological average, you've pooled equal amounts of all the samples together, then if you have linear quantitation of all of your other samples, right, then it should match the mathematical average, right, that you get from averaging all of these points, right? So this is the global average here. And you can see the study pool QCs for this analyte is laying very close to the average value that's measured for those samples. So what that tells us is that when we're comparing this quantity to that quantity, we're in the linear dynamic range of the instrument, okay? All right, so then we can perform statistical analysis, and not to harp too much on this, but we can pick some ions. We can use tags to tag molecules and of importance to use in many different comparisons. We then look at our principal components and how they're separating. We can find statistically significant molecules. Okay, so after we've done this, we like to try to get a putative identification of these molecules. And one of the really nice things about Progenesis is the ability to include a number of different search algorithms. And so import and utilize search algorithms. we tend to use lipid maps, either the full lipid maps database or a subset, phospholipids, and do either theoretical fragmentation or use database fragmentation. We've also used Lipid Blast. So what I want to show next is the picture again of significance versus the number of putative IDs. And I know for early, this is a lot of dots, right? This is a lot of dots to show so early in the morning. So what we're looking at here is just a difference in the same data, except on this plot, I'm showing the color as the significance. So anything that's red is basically above a Bonferroni significance threshold. Over here, what we're looking at is the number of identifications of those compounds. So if we just kind of filter the data and only look at the ones that are 0.01 and better, and then look over here, we can see that database searching is still a limitation within metabolomics. And I don't think this has anything to do with the particular software that we're using here. I think it has to do with how database searching is currently being performed, the number of molecules that are in there, being able to match fragmented, in-source fragmented to database searches. So we're very happy that we're getting a lot of these compounds that are significant with putative IDs, but we also have a number of compounds here, and it may be because I'm not searching the right library, it may be that my library doesn't contain the compounds of interest, but I just put a note in that I think that this is one of the areas where we're seeing the most evolution most quickly in the lipidomics literature is the ability to how do we do these proper identifications. It is, as an example of that, important to take a look at alternative identification. So we're doing a lipidomics experiment, and you can see some very significant changes here. This one on the left is from the experiment I'm showing, obviously highly differentiated between, and this just happens to be the drug that we actually used in the sample. So we were looking early eluting compound, unknown, check the mass. Oh, this is the drug that was actually doped into, it was dosed those samples. In the clinical study, I'll show us another slide in just a minute, we had, it was a sepsis biomarker study. These patients were really sick, and we actually find propofol in 5 out of the cohort of, I think it's about 80 patients. So that's another thing, maybe searching when you're doing human samples, searching a drug database is important to make sure you're not flagging as biologically significant molecules, things that are exogenous. Okay. So once we get the identifications done, the point I want to make here is that groups of putative IDs can be quite complex. So here, what in very small font again, is that we have a lipid maps matches, we have Progenesis Metascope matches from lipid maps again, but we also see that we've got a couple of different isoforms of the same diacylglycerol, right? So we're basically we're using this to assign the chain length. We've got ASMS spectra, pretty happy with this match, but we have to understand which one of these is right. And so one of the things I was talking to Mark about, excited to hear that we're moving towards only using the information that we have to call an ID. So many times you can call number of carbons and chain length, total number of carbons, and unsaturation with a lot of confidence, right? So we're moving towards being able to basically call that as a definite. We know the class, we know the number of carbons, we know the chain length, and the double bonds, but we may not necessarily know the exact unsaturation location or the chain length of each chain. So I'm excited to see things kind of moving in that direction. But here we make some putative identifications, and then we hand the data off to our collaborator, and we say the things we have MSMS data on. We're going to give you that information. We feel very good about that as well. We're going to run some fatty acid standards, and the manuscript goes in. Okay, so we're happy. So the first thing we get from reviewer 1, and this is airing a little bit of our dirty laundry here, so forgive me for this. So overall, the manuscript's well written. The studies are thorough. Combining the use of selective inhibitors with lipidomic profiling and correlating is important in the understanding of mechanisms. And I'm like, yeah, our stuff's important for this. We want our collaborator to think that. And then the wheels start falling off. But this reviewer has some major concerns, primarily about the lipid profiling. The authors should note how they derived their metabolite IDs. So apparently I didn't do a very good job describing that in the manuscript when I was writing that section. And at least one lipid in each class as identified of changing needs to be more closely identified. And then the diacylglycerol species are unusual and relatively minor, such as, and then they live some odd chain, really weird diacylglycerol species. And I'm reading this, and I'm like, where did these, I didn't identify these guys. Like, I didn't say that, right? And I go back and look, and sure enough, they had changed the table before submission, right, to basically pick the actual full sequence that was reported from the full report from lipid maps, right? So when you pull up a structure, there's no 32 0 for a 16 0 16 0. They always claim, right, that it is the full sort of full lipid map's name. So they had inserted just one random one out of the table of many that we had given, right, and reported that as the identification, right? So claiming more information than. So we felt really good, and then the wheels fell off, right? So how do we address this? So we go back to this piece of the pie here, and we offer quantitative verification in Skyline, and we had done this before, but now we've gone back. We run standards, okay? So what this does is it allows us to, and this is the data for the ceramides. We order a bunch of standards for ceramides. We analyze our data, so the standard, and analyze our data with those. We see QC pools, no drug and drug treatment, look at quantitative differentiation, and now this file can be shared with the world as far as what we are claiming for quantitative data. So we upload these files as supplement to the manuscript. So targeted analyte extraction, raw data verification, and publication of your results. We kind of can use this to verify. So this is another one, thiacylglycerols, quantitative information, raw data, peak view at the MS1 level. All right, so now what we've done, we go back, we run a bunch of standards, right? We confirm whether or not they're in this data, what the ionization form was of the standard, retention time error, whether we observed it in the sample and the supplier. So this is a different view of where I wanted to finish and where I felt confident, but where the reviewer required us to go back and actually, be more confident about our identifications. And so it's nice to kind of be able to add that into the pipeline and include that step. Unfortunately, we're not able to verify all of these because not all standards don't exist for all of these diacylglycerols. But we were able to convince them that since the number of all of the identifications that we had made putatively from Progenesis that we had standards for was correct. The fragmentation pattern matched for the others. The paper was accepted, and they had no further problems with the identifications. Okay, so switching gears a little bit, I want to now just show you very briefly what this outlier detection looks like on a much larger study. So this is a biomarker study looking at a sepsis cohort. And so we have a bunch of individual samples, the global average of those samples, right, and then our study pool QC for this particular lipid. When we analyze all the data, we see that within this sort of cloud, we have 3 outliers, biological outliers, sample outliers. So we need to quickly flag those, remove them from the experimental design. The nice thing about this is we don't have to redo our alignment or anything between the samples. We basically just go back to our experiment designs tab, remove those samples out of the design, rerun the PCA, and we see that our study pool QCs are in the middle, and now we've got essentially no outliers. When we overlay, this was a sepsis cohort. Half the samples were from Cambodia and half from Duke. So when we look at that, we can see that within this cohort, the Duke samples are in orange, and the Cambodia samples are in purple. And so we have a spread between the means of these. And so out of multiple plates of samples that were run to do this study, they found that the bias that was in this was pre-analytical, probably due to sampling location and shipment, potentially from Cambodia to overseas. Statistical analysis yields really important differentially expressed ions, and so now we're undergoing the process of identifying these and confirming them with standards, those things. So the last thing I want to cover is, and I mentioned earlier, the importance of DDA in data in lipidomics analysis. And the reason I mentioned was the fact that we like to have isolated precursor information. And so I think everyone is familiar with data independent acquisition, whereby selected bins are isolated in the MS2 space. So like swath, right? We'll use a 12 Dalton bin or a 20 Dalton bin, right? And then they move to the next bin, move to the next bin. So some of our colleagues at Waters have come up with a really nice alternative to that, and that utilizes the ion mobility drift data acquisition strategy, but instead of moving a quad in a fixed momentum, it actually sweeps the quad from low to high. So what it does is basically what you have is you have a quadrupole transmission window, and instead of moving it in a fixed windows, it sweeps it. So if you just do a little thought experiment, understand why this is important. So if you're sweeping this quadrupole from low to high, and this is called Sonar, and Waters is calling it Sonar. I have no idea what Sonar stands for. So what it does is when the edges of those compounds, think about for an MS imaging experiment, the people are able to get oversampling because as they move the sprayer along, it's larger. You get better resolution than you do the size of the bin. Okay, so as this is scanning from low to high, the edge of that bin appears and then disappears. Basically, right? It appears there and then disappears. So even a compound which has a mass which is much closer will appear slightly earlier in the scan, right, and disappear slightly earlier. Okay, so what they're able to do is even though they have an isolation width of about 20 AMU on the Q1, they're able to narrow the location of that MSMS spectra within about a Dalton. Okay, so by sweeping the quadrupole, they're able to do DIA with about a 1 AMU resolution on Q1. Okay, this is also extremely fast. The total scan time is less than 0.2 seconds. So that's very important for us when we're doing lipidomics application, where our peaks may be anywhere from 4 to 6 seconds wide, maybe even narrower in some cases. So we're able to do really high duty cycle separation. So this is just going over why we do low energy and high energy collision energy. So we get fragmentation here, and then we get MS1 information. So another way to look at this is so when we're sweeping this quad, we get this relationship between what's effectively the bin, right, the drift bin, or for the acquisition strategy. So this is looking at data in Skyline, right, and mass to charge. Scanning over, this is a peptide separation. So we can also then just isolate what the data looks like. So this is the MS1 spectrum. We can isolate what the data looks like in that bin, cleans up our MS1 spectrum. Same for MS2. You can isolate what looks like in that very narrow MS2 bin. Basically cleans up and gives us a nice clean MSMS spectrum. Okay, so even though this data is being collected in a DIA format, it gives very clean MSMS-like spectra. This is an example from Hans Visser's that Hans sent us. So this is basically what they're doing is they're doing a lipidomics separation. At this point, they've got a 10-second at base or 6-second at half-light peak width, and they're able to get 24 low energy and 24 high energy scans across that peak. Okay, so what we're enabling by doing this technique is data-independent analysis on a very, very fast time scale with DDA type MSMS spectra, which I think is very exciting, both for quantitation on the MSMS ions and for qualitative confirmation. So with that, I'd like to thank my collaborators. I'm not sure who MI is. I think that was a typo at the end of last night. But Arthur Mosley, Laura Dubois, and Eric Soderblom, Matt Foster in our lab, Brian Pratt, Brendan McLean, and Bhagisha Sharma, especially for all their work on Skyline and Panorama. And then Waters, too many to mention, but particularly for this presentation, Jim, Hans, Keith, Jason Wildgoose, and Chris Hughes. Thanks, Mark. Thank you very much.
Thanks.
17 minutes, Martha.
Maybe time for one quick question?
What is the cutoff used for the CV? What percent?
Yeah, so it kind of depends on the application, but typically we like in a discovery experiment, anything less than 20% CV across our pools, we don't even consider it for statistical analysis. 20%. So 20% in a label-free type amount where we don't have standards for everything, right? We're just looking at peak intensity from run to run to run. 20% is what we typically. There's still plenty of candidates typically at 20% CV. The average CV for these experiments is about 7%. So between the study pool QCs from, across, in the last study, the biomarker study, we ran nine pooled samples over a period of several days, right? And the CV maybe a little higher, on average about 9%. 90% of the data is less than 20% CV.
If you look at the publications, it varies quite a bit. Someone uses 30%, 20%, 15%.
Yeah, there's no standard. I think that's a good point. What do you use? I'm interested to know.
I was using 30%. That's what I'm asking you.
Yeah. So I think that's probably on the top end of what we would consider. If something was really statistically significant, large fold change, we might consider it at that. Typically, when the CV is high, it's because of low abundance, right? The CV, and so you have to consider whether or not it's and here again, that's another reason that Skyline comes invaluable is because you can really take another look at the raw process data as opposed to any kind of transformation that has to happen to look at the data. Skyline, they don't really do any, I mean, McCoss doesn't believe in any kind of peak smoothing, any kind of data transformation. It's really a different strategy, right? So they just look at the raw data. And so at that point, I would probably, if I had a really promising something at 30 or 35%, I'd want to see the raw data.
Thank you.
Cool. Thanks again, Will. And we
Okay,
can detect some more questions at the end.